Our last session here is being run by Ryan Spitler here at the PHIND Center. Dream team, where are they now? Ryan is Deputy Director of the Precision Health and Integrated Diagnostics Center here at Stanford. He previously served as Deputy Director of the Stanford Canary Center for early detection of cancer, and Program Manager and Chair of the Operations Board for the International Alliance for Cancer Early Detection. Ryan got his PhD in cellular and developmental biology at UC Irvine, and he's been a partner in crime with me here at PHIND since I started. Ryan, why don't you come on up?
Thanks, Joe, and thanks, everyone. Yeah, so I think, part of this, as we get towards the later part of our day, I kind of want to reflect on some of our origins. And, in the beginning, when Sam and I launched the center back in 2017, we also launched some really pivotal programs, and we looked for really inspirational groups of people working. And what we called those teams were dream teams. And so these are teams that started in 2017 with us. And now, several years later, they've been really excelling and doing really great work and sustaining this effort in really meaningful ways. So we really wanted to look like where are they now, where's that's coming from, and really showcase a lot of the exciting work that they've accomplished over this time period. So I'll start out by introducing Dr. Ian Gotlib, who is a professor in the Department of Psychology and director of the Stanford Neurodevelopment Effect and Psychopathology SNAP laboratory at Stanford University. Dr. Gotlib examines psychobiological factors that place children, adolescents, and adults at risk for developing depression and engaging in suicidal behaviors, as well as processes that are protective in this context. So really looking forward to hearing from you, Dr. Gotlib.
Great, thank you all, and thank you for inviting me. I guess I can just walk, right? Great. So with the rest of I know I'm presenting today, but Holden Maker and Rachel Manber and Dennis Wall, all part of this. We're so appreciative of the support that we've had from PHIND. And what I'd like to do today is to talk a little bit about the context for this PHIND funding and what we've been able to do with it, where we're going, some things that interrupted our research, some things that made it actually more interesting inadvertently. So I want to start by talking about depression and suicide. So welcome to my life. I want to try to emphasize to you just how prevalent and important these two constructs are. So depression is the leading cause of disability worldwide. And when we talk about depression, we're actually talking about a psychiatric diagnosis. I know that many of us can wake up some morning and say, oh, I'm depressed today. We rarely wake up and say, geez, I'm schizophrenic right now. And I want to convey to you that depression is as burdensome, as disturbing a disorder as schizophrenia, if not more so. So 300 million people in the world diagnosed with major depressive disorder. It has an enormous $500 billion cost associated with it in terms of direct treatment, lost work productivity, absenteeism. It's also a recurrent disorder. So 80% of people who are diagnosed with a depressive disorder will have another diagnosis, another disorder episode in their lifetime. And so if we can prevent that first episode from occurring, it's, I think, the only way that we're going to make any kind of dent in the prevalence of this disorder. It's also a risk factor for suicide, a 2 to 3 times mortality risk for depression. The problem of suicide is, as we'll talk about in a moment, increasing. There are almost 1 million suicides per year at this point, 40,000 of them in the United States. Twice the prevalence of homicide, 5 times the incidence of HIV, AIDS. And I want to just show you what we've been able to do and not do in health. So you can, oh, okay, interesting. So you can see that we've made tremendous progress in reducing prevalence of a lot of medical disorders like AIDS and stroke and heart disease. And we have done nothing with suicide, that the rates of suicide have just stayed constant, and some people will argue have increased over the last 20, 30, 40 years. Adolescence in particular is the highest risk period for both depression and suicide. So first episodes of depression and suicide often occur during adolescence. And at this point, we estimate that about 1/4 of all adolescents will experience a diagnosable episode of depression. Okay, 60% of adolescents who commit suicide have had a previous depressive episode, and suicide is the second leading cause of death in adolescence. I know that shouldn't be surprising, given that adolescents tend not to die of things other than accidents, but suicide is a very close second. And I want to show you that these rates of depression and, as we'll see, suicide have been increasing over the past few years, and particularly in females. So you can see rates of depression going up from about 2010 on, and these are data until, 2020, so just 3 years ago. And they've been increasing enough that there are articles everywhere about how rates of depression, rates of suicide are increasing in adolescence, enough that the American Pediatric Association suggests that all teens be screened for depression. Suicide rates climbing, right? Just 3 months ago, the Centers for Disease Control put out their Youth Risk Behavior Survey. And I just want to quickly share some data, because these are as current as data are going to be. And you can see that persistent feelings of sadness, 2021, almost 2/3 of adolescent girls are reporting feelings of sadness. A third of them, have considered attempting suicide, and 13 have made suicide. 13% of adolescent girls, twice as many as adolescent boys, have made suicide attempts. Some of this is COVID related, and we'll talk a little bit about this, but this predated COVID by a lot. Why aren't we stopping this? Why aren't we being able to do really much more about this? Well, a lot of the reason has to do with limitations of previous research. So a lot of previous research are single point cross-sectional assessments, often based on subjective, how are you feeling reports with small samples, and typically only a single risk factor. Let's see if early stresses predicts depression. Right? So depression is multi-determined, as we'll see. And what we wanted to do with PHIND is to see if we could integrate data from multiple domains to see if we could identify those children at particular risk for developing depression and engaging in suicidal behaviors. If we can do that, our belief is that we had the potential here to prevent that first episode of depression and this kind of lifelong negative cascade. So I want to describe how we came into PHIND with Ryan, with Sam Gambier, when Sam developed Precision Health here. So we had recruited and assessed a sample of about 200 boys and girls ages 9 to 13 who had been exposed to a range of early life stress. And prior to PHIND funding, we had actually conducted 2 assessments of these children 2 years apart, okay? When they were 9 to 12, and again when they were 2 years later, around 11 to 14. And you can see it here, right? When you look at increases over adolescence in depression and suicidal behaviors, this was what we had done when we came to PHIND. What we had proposed to PHIND is that we continue to assess these same kids, but now during the period of increased risk for depression, increased risk for suicidal behaviors. And based on epidemiologic data, we expected that about 25% of this sample that we would see them start here, engaging in depressive ideation, suicidal ideation. And so with PHIND funding, and I'm just going to focus here on 2 of the things that we did. We looked at inflammation, that is, we drew blood from these children, and we continued to do neuroimaging with them. So I'll talk a little bit about cytokines and C-reactive protein, but we wanted to see if we could get inflammation markers prior to the emergence of depression, prior to the emergence of suicidal attempts, suicidal behaviors, to see if we could predict which kids would engage in these kind of maladaptive behaviors, and do the same with imaging. I will tell you now, of course, imaging is not particularly scalable. These are finger prick blood drops, and that's absolutely scalable, and one of the things that I want to talk about. So we had found that early life stress, these kids were exposed to stressors. The more stress to which they were exposed, the higher their levels of internalizing and externalizing, sadness, anxiety, fears, depression, and acting out, particularly in girls. So we could have a sense of which kids were going to start engaging in maladaptive behaviors. But with PHIND funding, we were actually able to predict diagnoses of depression and engaging in suicidal behaviors. And let me just show you that we could predict a subsequent diagnosis of depression from IL-6 and TNF-alpha, so from cytokines in their blood. So we're able to use inflammation data to predict which kids subsequently are going to exhibit depression and engage in suicidal behaviors. We can do this with suicidal behaviors as well, again from IL-6, TNF-alpha. We also can start to predict which kids are going to exhibit depression and suicidal behaviors by their patterns of brain activation. And in particular, those children who were less responsive, less reactive to reward stimuli before the onset of depression, right, as they were still at time 1 and time 2, were more likely to have higher levels of depression, higher levels of suicidal ideation, right, with less activation. And it's not just brain activation, it turns out it's brain structure. So if you look at regions in the brain that are associated with reward, like the caudate and the nucleus accumbens, you find that lower volumes in these structures predict higher levels of suicidal ideation. So we're able to take these measures before the onset of a disorder, exactly what Sam, I think, had envisioned, and predict which children are at greatest risk for exhibiting these kinds of maladaptive behaviors. We also, having this sample, started to branch out a little bit. And we started to look at the broader environment, and in particular, neighborhood disadvantage. So children living in relative poverty, children whose parents had lower levels of socioeconomic status. And what we found is that those children living in the areas of greatest neighborhood disadvantage were exposed to the highest levels of early life stress. Perhaps not surprising, but those levels of neighborhood disadvantage seem to be related to a concept that is called biological aging. That is, the more disadvantaged their neighborhood, the faster they were aging biologically. We have a chronological age that's fixed. You don't want to know how old I am, but we have a biological age determined by telomere length of caps on DNA, by different patterns of brain reactivity. Now I'll show you that these kids who grew up with the highest levels of neighborhood disadvantage had the shortest telomere length, a marker of stress. Okay? They also had the strongest neural connectivity between prefrontal and amygdala areas that shows biological aging. They're aging faster than their chronological age as a function of higher levels of neighborhood disadvantage. Okay. So, what do we have? We have adolescence as this period of risk with early life stress, predicting levels of depression in general, but also with PHIND funding, really being able to focus on the impact of biological aging, of what we think are probably mediators of this association, inflammation and neighborhood disadvantage. I just want to share with you, I've got just under 5 minutes, and I won't go long, I promise, but I want to share with you something that you may have thought was coming, but we didn't. And that was halfway through our COVID funding, COVID happened. And Stanford was completely shut down. And we were shut down for about a year. And remember, we were bringing in kids for blood draws. We were bringing in kids to do brain imaging. That stopped, right? And we couldn't do any of that. So during the shutdown, and you all remember these newspaper articles, right? We started sending out surveys, which is all we could do, online surveys to kids. And not surprisingly, you see these high levels of distress in kids during COVID. I mean, we found it, other people have found it, and the greater the early life stress they've experienced, the more distressed they were during COVID. It seems to be, again, a stress story. For us, though, the more interesting thing happened when we were able to resume a year later. These kids, we asked them if they would, do you mind stop growing? Do you mind stop aging? And so we brought them all back and we continued our study. And one of my concerns is that these kids now, post-COVID, post-lockdown, are not the same as kids were, as they were before the lockdown. So what I want to present to you is, in answer to that question, are 15-year-olds after the lockdown just the same as 15-year-olds were before the lockdown? Are 17-year-olds the same? And I'm going to suggest to you that they are not. So we ran about 100 of these kids now after the lockdown, but we also had data before COVID. Remember, we had started this almost 10 years ago. So we have data from kids pre-COVID. And so we were able to compare same age kids that we assessed after the lockdown ended to kids that we assessed before the lockdown. Completely different kids Does that make sense to you? Okay. A 15-year-old before COVID with a different 15-year-old now. And not surprisingly, again, much higher levels of depression, of anxiety, of kind of sadness and fear. But what surprised us is that their brains are different. That after COVID, these brains, children's brains are older. They aged what looks to be 3 to 4 years in that 1 year of lockdown. So greater cortical thinning, greater hippocampal growth, everything that's associated with early stress in other samples, we found that's how they reacted to the pandemic, is these faster biological aging of the brain. So inflammation, also much higher levels of inflammation post-COVID, right, than they had pre-COVID. So I'm just gonna skip for a moment. So you remember this. And I just wanna present you with two different scenarios. And we don't know what, so COVID is here, right? So here's one, which is what we're finding, right, is that these rates continue and just are off the charts. It's also possible, though, that it's this, right, that the lockdown's over, COVID's largely receded, maybe this is a blip. And because we were able to do what we did through COVID, we're now able to bring these people, these participants back, all of them at age 20, and repeat this. And so we'll be able to determine whether it's this or whether it's this. And we could not have done that without PHIND funding. And so we're able now to identify kids at risk who are going to develop depression. COVID, we thought, was just gonna mess with all of this, but it's actually given us some interesting data. So just that zero, Ryan, thank you very much. And again, we could not have done this without your support.
Great, really fascinating work from Ian Gotlib. Next, I'm excited to introduce Dalia Perelman, is a registered dietitian and certified diabetes educator. She has always been interested in both nutrition research and its application in clinical practice, and has worked in both environments. So, looking forward to your talk.
Hi. Thank you for sticking till almost the end, and I hope it's worth your time. I'm part of a beautiful dream team. I'm the dietitian in the team, so don't ask me too many questions about algorithm development, etc., but I can tell you all you want about diet. And I work with Mike Snyder, who is he's in the genetics department here, and Tracy McLaughlin, who is an endocrinologist, and we have such a great team. And I want to showcase some of what we've done with the help of the PHIND funding that really got us going. And every postdoc that comes in is like, oh, can we be part of this study? And we keep adding and adding stuff to the study. And now it's like a mammoth thing that I think no one can fund because we grew too much. So, all right, let's see if I can understand how this works. Okay, so I'll talk about phenotyping of diabetes or glucose dysregulation because it's bigger than just diabetes. I'll talk a little more about how what we eat affect us and our future plans to learn about how when we eat has a huge impact. I'll spend a little more time on the first topic because that's the one we've developed the most, and the other two are just glimpses of where this project is going. So, type 2 diabetes, I'm sure, I don't know if I'm the first one showing this today or you've seen it ad nauseum. But yes, it's a big problem. Diabetes, it's a big problem. About 11% of Americans have it. And we defined it by your levels of glucose, either at a fasting level. So you go to the doctor fasting, they measure your blood sugar. If it's above 126, you have diabetes. if they can measure it via an A1C, which is an average of your blood sugars over the last 3 months. And if it's above 6.5, you have, how does this work? How did you make this work?
Very top one. Hold it down. No, the very top little bit.
My finger doesn't work. Well, maybe this. There we go. I can use both sides of my brain. Okay, so 6.5 or above, you have diabetes. We can also do this, which not too many people love doing because you have to sit in the lab for quite a few hours. You drink a delicious drink of 75 grams of pure glucose. They measure your blood sugars 2 hours later to see how you dispose of that glucose. If it's 200 or above, you have diabetes. So those are the ways we can measure diabetes. 1, 2, or 3 of these, you have diabetes. So that's how we define it. But truly, the physiology is very different among us. So maybe, different people have different types of diabetes. So that's what we were trying to figure out. Prediabetes, as I showed you in the previous slide, is the levels in between. So normal, prediabetes, diabetes. And a lot of people have prediabetes. It's a little scary because it's about a little over a third of us have prediabetes. A lot of people don't even know it. And a lot of people will go on to develop diabetes. So this is bad. And when we were thinking about this, we said we have to intervene somehow and make a change because these things are getting worse. So once you have diabetes, and actually what we've learned a lot is that the complications start very early on. They start very early on. So, and there's all over the body. You can see the brain, the heart, the kidneys, the nerve endings, the mouth, everywhere. Why? Because it's in your blood. And the blood goes everywhere. And it will affect different organs differently in each one of you. So some people might develop retinopathy, which is problems in the eye. Some will have mostly kidney issues. Some will have cardiovascular issues. Some will have all of it. So it's definitely something we need to work on to try to prevent. So don't get nervous. We'll walk through this. Okay, so we want blood sugar to be kind of stable. We like it to be somewhere, I would argue, between 65 and 130, 140. Somewhere in there, it's a good level. Blood sugar is going up and down. You have a meal, and you're eating something. Maybe it has some carbohydrates like rice or ice cream. And that actually in your intestinal tract, you have hormones, we call them incretin hormones, that are already going out of the intestine to tell your organs what's coming. Hey, there's some protein. Hey, there's some glucose. Get ready. So the rest of the organs are getting ready to make sure you can digest and absorb what's coming through the digestive tract. Those incretin hormones are going to the pancreas. It's one of the things they do among many others. They go to the pancreas and they tell the pancreas, hello, beta cells in the pancreas, start making some insulin because glucose is coming into the blood. So even though glucose has not reached your blood at that point, it's still in the stomach, intestine, somewhere in there, the pancreas already has been alerted to start making insulin. So by the time glucose is out there, it's ready to go out. So pancreas secretes the insulin. The insulin helps your liver get up some of that glucose to store it as glycogen for the lean times. And the rest, the muscles are happy and taking some glucose. The fat cells are happy and taking some glucose, to store it for leaner times. So all of those things help. Blood sugar comes back down. Normal levels. Everything's working beautiful. You stop eating. Some of you might be fasting. Some of you might just go to bed and not eat through the night. And what happens? All the organs need to be fed. So the alpha cells in the pancreas are secreting this glucagon. Glucagon goes and tells the liver to break down the glycogen that it's stored up here release it into the blood, and help us maintain a stable blood sugar. So as you can see, there's a lot of organs that are involved in helping keep our blood sugar in good control. So there's the pancreas, there's the intestinal cells secreting the incretin hormones, there's the muscles and the fat cells taking in the glucose that's in the circulation, there's the liver having a tremendous role. And I didn't talk much about the kidneys, but the kidneys also have a role in helping you either release the glucose out into the urine, or if it's a healthy kidney, it reabsorbs the glucose back into the circulation. So lots of organs involved in maintaining the glucose homeostasis. And this is what we've been taught, this is what we see in many of the patients, is your blood sugars are normal, and then what happens is you become, some of us become insulin resistant. So what does insulin resistant mean? The cells, the muscle, the fat cells are resistant to the action of the insulin, and they're not taking the sugar into the organs. Glucose stays in the circulation. So as that is happening, initially the pancreas says, oh goodness, there's a problem here, I need to secrete more insulin because the cells are not listening. So the pancreas over-secretes insulin to compensate for that defect, and blood sugars are still stable. Beautiful, right? Our bodies are amazing. So you develop insulin resistance, but compensate with extra insulin production, blood sugars are stable. Until the point where the pancreas is like, okay guys, enough of this, it cannot keep overworking, I'm burnt out. And starts to secrete more normal amounts of insulin, cannot compensate for the insulin resistance, and we see blood sugars increasing. So that's what the thought is, and that's what we've seen in a lot of people. But interestingly, this is what some of the findings that we've done when we started doing this study. So these are people, we organize them here by A1C. So all the blue ones have normal A1C, so these don't have diabetes. These are people with normal blood sugar control. These are people that have prediabetes based on the A1C measurement. And we had one person, we were not enrolling people with diabetes, but this person didn't know they had diabetes, and we just like, okay, just go through the study. So as you can see, there's insulin resistance all over the place. It's not separated by those who have prediabetes and those who don't. I mean, initially we thought this would also look like that, like an increasing line of deficiency. And no, it's all over the place. And the same with beta cell function, which is, we're measuring by how much insulin it's producing, and incretin function. Everyone has their own journey. So this person here maybe goes to the doctor, they say, oh, you're perfectly fine, but they have a lot of insulin resistant and some incretin dysfunction. They might be at higher risk of developing prediabetes and later on diabetes. These two people with the same A1C are looking very different. So you can see how heterogeneous this is. It's not so clear-cut. So what we thought is, if there's a way to do this and target the treatment, there's very targeted treatments for each one of these dysfunctions. We're not over-medicating people because we don't need to do this polypharmacy to eventually find something that works. We're giving them more, it's more precision health. So, precision medicine. So, why don't we do that? Like, okay, just do it, right? Well, it's really hard. It's really hard to measure. So the insulin resistance, at this point, we're measuring with this test that we call the SSPG. You have to go, at this point, only is done in research settings. And you sit there for 6 hours with infusions coming in and out. I'm looking at the clock, so I won't go through how the mechanics of the test. If you're curious, I'll let you know. But you're sitting there, infusing insulin, infusing glucose, stopping your pancreas function for a little bit. It's a very, very complicated test. Not everyone can do it. And you can imagine that's not something we can translate to the clinic. How do we measure if beta cell function? Is mostly with an OGTT, the oral glucose tolerance test. But the test that we're doing in the clinic is 3 hours, not 2. And we're taking 17 samples that you're sitting there, not 2 or 3 as you do in the clinic. So again, not very translatable. And incretin function, it's a fun test. Because how can we test that? And we test it via a very, very fun way, like in indirect way. This is when you drink the glucose, your blood sugar goes up and then comes back down as all these organs are doing their job. Then when we do this IIGI, which is intravenous glucose infusion. And how do we do that? We infuse glucose, trying to match precisely the glucose curve that you had when you drank the glucose. So your body is exposed to exactly the same amount of sugar in your blood. We're just bypassing the digestive tract. So when we see the difference in how much insulin is produced when you drink the glucose versus when you get the glucose via an infusion, we can see this is the incretin effect. So the farther apart these two lines are, this person has beautiful incretin effect. This person has no incretin effect. So this person has a problem there. This person does not. So you see how complicated it is to get at this data? It's like, oh my God, I need to devote 3 days at this research center with all this staff involved, and not very translatable. So what we thought is, look at those OGTT curves. I said we take 17 time points. So we're going, maybe the shape of the curve, how fast it goes up, how fast it comes down, how long does it stay up, how, all of the dynamics of the curve are telling us something about the organs. Oh, it's here. Oh, that's great. So maybe the dynamics of the curve will tell us something about how the organs are functioning. So this amazing postdoc, Ahmed, he extracted all those features, the peak, the area under the curve, the slope, the curve size, a lot of stuff about that curve. And he trained it, and he did some machine learning on that to predict the metabolic phenotype. So he did that with the data, and he did that with Z normalization, which is smoothing and taking out outliers and things like that. So he did it different ways, but playing with the shape of the curve. And what we found is that using that, these are the orange and the pink one are our OGTT features. They perform beautifully to predict muscle insulin resistance, beta cell dysfunction, impaired incretin effect, and it's much better than what we have up to now. So, wow, we have a way to do this. So, how about someone goes to the doctor says, here, take a glucose, take this glucose drink, wear this CGM, we'll look at your curve, we'll determine what subphenotype you have, and we can then personalize the treatment to you. This is translatable to the clinic, and that's what we were working towards. So we have participants both coming to the clinic to do the OGTT, to get the venous blood, and we give them a CGM to do it at home, and we're playing with those numbers, and it's working out really, really well. So that's one of the exciting findings, that we can now start thinking about translating that to practice. Now, about what we eat. So this is a study that was originally funded by PHIND, and all I've talked to you about is just using the information that we got from baseline. So the OGTT, SSPG, IIGI. We also took a ton of omics. So we're further working into finding biomarkers and other interesting associations there. But we also tried to play with their diet and giving the participants different types of food to see which one they react differently. Again, we want to be more precise in our recommendations to participants. So we gave them these different types of carbohydrates, exact same amount of carbs. They are all getting, they're all getting 50 grams of carbs in the different foods. So beans, berries, bread, grapes, pasta, potatoes, and rice. As you can see, some people react more to one and to the other. So we're trying to understand, can we predict that using maybe the microbiome samples, maybe the metabolomic samples, maybe the proteomic samples. So something so we can be a little more precise in telling people what they can eat, what they need to be more careful with. And we're still playing with this. Yue is now owning this part of the study and trying to marry this with all the omics. But then the interesting thing is that we're also giving participants rice with protein, with fat, or with fiber to see if one of them attenuates their response to glucose. And we indeed saw, for example, this person fiber, which is the red one, really helped to attenuate the peak of rice, which is the blue one. But the other things didn't help as much. For example, protein didn't help as much. The orange one didn't help very much. This person, everything helped. Rice by itself didn't work. Just put the rice with protein or fat or fiber, beautiful response. So as you see, even that is personalized. And we're trying to figure out what determines what and who will react to what. So that's still in the works. All these amazing postdocs working their magic with all these big data sets. And I'm sure next time I can talk a lot more about this. And lastly, in 1 minute, Hey June, who is another beautiful postdoc in the lab, she's looking at, okay, these people, when they put the CGM to take all these funny meals we give them, we have a lot more data about them. We have all the data, everything else they're eating through the day. They're also wearing a Fitbit, so we can look a little bit at their sleep. We can look about their physical activity, and everything is time-stamped. So we know when they do what. So she is building an algorithm to try to understand glucose outcome based on everything we know about these participants. All their phenotyping, all their clinical labs, all their CGM, and all of these things. And she's coming up with amazing, beautiful associations that we can then give to participants and tell them, this is what works for you and for this reason. So as you can see, people eat, this is percent of calories and at what time of the day they're eating them. Different people eat, they like to eat at different times. And she's finding out some interesting things. So this is my last slide. So eating later in the day is associated with higher A1C, insulin resistance, and incretin dysfunction. So when you do intermittent fasting, at this point we're saying, well, maybe skip dinner, not breakfast. Sure, very doable, right? But that works for your metabolism. Insulin-sensitive people derived more benefit from increasing activity in the afternoon, and the resistant people better off when they increase activity in the morning. So maybe something about the insulin resistance versus insulin production is going on in here. Walking before bedtime helps everyone for the next day's glucose levels. And we have beautiful clues that sleep quality and duration have a very, very strong association with blood sugars. So what we're doing in our next study is we're putting them on even more devices. So they're logging their food, they're using a circle ring which will measure oxygen consumption so we understand a little bit more about how they're sleeping. A dream band, which is it's basically like doing a sleep study at home. So we're going to measure their sleep, their blood glucose levels, and temperature, continuous temperature, because there's a lot of clues about your metabolism on those minute changes in temperature through the day, and the Fitbit. So if you want to be a little robot and wear a ton of things and test a lot of things, and we're changing the timing of the meals and changing the distribution of the calories so we can continue to understand how all of these different variables work together for each one of us to predict our more, the healthiest way to maintain our blood sugars. So with that, thank you. And no, I'm a little bit over, sorry.
So next we have Dr. Anthony Wagner, who's a professor in the Department of Psychology and Deputy Director of the Tsai Neurosciences Institute at Stanford University. His translational research examines aging and Alzheimer's disease, the relationship between multitasking and cognition, and the implications of neuroscience for law. So, welcome.
All right. It's great to be here. I view this sort of presentation as both a thank you and a celebration of those who are on the SAMS team of what we've achieved. So what I'm going to try to do, let's see if we can. So the Stanford Aging and Memory Study, it's a study that started out trying to understand both moment-to-moment differences in brain function, as well as individual differences in brain function that underlie differences in memory, both in the moment and between people. And thanks to PHIND, we've now extended it to be a longitudinal study to try to understand changes in brain function, in particular memory function, over a 7-year period. And so I'll describe how we got there. But first, I'm just one person up here as part of a broader team. I'd like to first acknowledge Beth Marmino in neurology, my co-lead on the SAMS project. None of what I'm describing would have happened without Beth. The PHIND team is a group of interdisciplinary scholars, additional scholars in radiology, as well as neurology. Carolyn is now at Yale, but remains actively involved. These are, of course, the sort of the PI members of the PHIND team, but we're a large group to pull off this science. Some of the SAMS team, I'm saying PHIND team, some of the SAMS team are here today joining us. All right, so the work in SAMS started off really, again, trying to understand memory and moment-to-moment changes in memory function, as well as individual differences in memory function in cognitively unimpaired, cognitively healthy, as we might believe, older adults. And one of the potential sort of drivers of variance in memory function, even in cognitively unimpaired individuals, is sort of preclinical AD-related processes. We know that Alzheimer's disease affects millions of Americans, affects millions of folks around the world. We know, well, there are 2 sort of drugs now that appear to have some modest impact on progression, amyloid-targeted drugs. There's no disease-modifying drugs that really change the arc of AD. And one hypothesis is, well, we need to sort of intervene, both develop intervention drugs, methods, but intervene on individuals during the asymptomatic period, well before there's been this cascade of neural and other sort of impacts on the brain. And so we need an individualized sort of precision prediction tool or approach to be able to predict AD risk in cognitively healthy older adults, asymptomatic individuals. And that currently does not exist. We again know that molecular pathology, network dysfunction, focal atrophy, they're present long before the cognitive symptoms that are the hallmark of mild cognitive impairment and then ultimately AD. We need sensitive tools that measure early functional and structural change, perhaps with imaging techniques, though there is the scalability issue, and combine these with other kinds of measures that might pick up on AD, evidence of AD presence during the preclinical stage. So in terms of episodic memory, it's the kind of memory that's impacted in Alzheimer's disease. This is memory for everyday life events. This is why the first typical presenting symptom, cognitive symptom in AD is the inability to build new memories for life events and be able to retain them over time such that you can remember that you had that conversation with a loved one previously and you don't repeat yourself, for example. We know that episodic memory declines with age. So the slope of decline is observed. It's steeper as we're starting to hit later in the lifespan. But importantly, in cognitively unimpaired older adults, about 30% of them carry these sort of these markers of preclinical AD, these amyloid and tau markers, be it from the CSF, now blood plasma, or with PET. And you can see that the rate of decline in episodic memory performance is steeper in individuals who are amyloid and tau positive, be it from CSF or PET. And in the SAM study, we also see this relationship between these AD biomarkers. Amyloid positive individuals who also have elevated tau, they're the ones who, in a cross-sectional analysis, they're the ones who are underperforming in terms of episodic memory relative to other individuals. And so one of the questions we have is whether amyloid and tau, how does amyloid and tau burden, how does it impact neural mechanisms that underlie episodic memory in older adults? We also know that E4, ApoE4 allele is a primary AD genetic risk factor, and E4 carriers demonstrate elevated tau, as well as they're more likely to be amyloid positive. And there's a number of possible pathways through which ApoE could impact the brain. One is through sort of this amyloid to tau cascade, but there are other possible sort of processes through which ApoE could actually impact neuronal health and memory function, including processes independent of amyloid and tau, including recent data suggesting that ApoE E4 might break down or impact the blood-brain barrier. So in SAMS, we're also interested in how ApoE genotype might relate to these fundamental neural mechanisms of memory function. And so what are we talking about in terms of these neural mechanisms? So if we think about a life event, a moment, an episode, you're having an experience right now, right? It's a multi-featural event. You're sitting here, your brain's representing this space, this environment, you're seeing my face, you're hearing the words, your brain's representing the sounds and translating them to meaning, your brain's interpreting the slides, et cetera. So events are these multi-featural experiences that are happening just in that one moment for your brain. These features are represented in different cortical regions. And we know that sort of cortical representation of event features are changing as we get older. In addition, these cortical features, these cortical representations of event features ultimately propagate their signal into the hippocampus, the structure on the medial or the inner wall of the temporal lobe. That's fundamental for building and later retrieving memories for life events. And there are specific computations that have been thought to be performed by this hippocampal circuit that are fundamental to learning. Following, if you've successfully built a memory and coded that memory in your mind and brain, at some point in the future, you encounter a retrieval cue. And a retrieval cue interacts with a whole host of things, including your goal, your attention networks of the brain. But the retrieval cues ultimately propagate into the hippocampus and are thought to trigger what's been dubbed pattern completion, a reinstatement of that hippocampal ensemble within the hippocampus that was built during the learning event. And that hippocampal ensemble then reinstates variants of the cortical patterns that were present during the prior experience with that retrieval cue. You can think of this sort of cortical reinstatement as essentially your brain traveling back in time, going through neural time travel, and reinstating, recapitulating, bringing back online these other features, this other information that was co-present with the experience. And that is the heart of, that is the conscious, that underlies your conscious awareness of remembering that past and those past event and the details of that past event. So in SAMS, when SAMS started initially, now 9 years ago or so, the grant wasn't framed, the project wasn't framed as a prediction project, as a longitudinal project. We were interested in integrating these functional imaging assays of hippocampal-dependent memory mechanisms with structural, molecular, and genetic measures to see if we can explain both within and between person variance in memory performance in cognitively unimpaired older adults. Our participants are remarkable. They came in easily in 3 full-day sessions spread out over a few months. And for some, they came in for 4 full days spread out over a few months. In addition to sort of blood, being able to do ApoE genotyping, we also collected CSF. And from blood plasma now, with collaborators, we're also able to sort of read out not just from CSF, but also from plasma, amyloid and tau, global amyloid and tau burden or status. But added to that, and this is sort of where our participants were heroic, they came in for a full sort of fMRI scan session. They were in the scanner for easily 2.5 hours or so. That's a big lift for older adults lying in an MR system. We're collecting data, measures of brain function as the individuals are building new episodic memories, and then we're also collecting data as they're being cued to remember, to retrieve those memories. And we're feeding those brain patterns, the brain data that we collect, into machine learning algorithms that can essentially allow us to get a quantification of the cortical representations that are being built during the experience, the reinstatement of those representations during remembering, as well as hippocampal function. We're adding to that not just structural measures at 3T, but ultra-high field 7T structural measures, 220 micron measures of the hippocampus that allows us to now get thickness measures and volumetric measures in particular subregions of the hippocampus that we and others have observed to be thinning in particular sublayers in area CA1, we've observed to be associated with worse episodic memory performance in older adults, so we're trying to go to scale on this. And then at the very end of wave 1, we also added tau PET imaging for a small subset of the participants. Our sample, the full sample in wave 1 is 212 older adults. They varied in age from 60 to 88 years old. Mean is about 70 years old. About 25% are amyloid positive, about 25% turned out to be E4 carriers, and all of them are CDR0. They're cognitively unimpaired. Not only do we have leveraged this CDR score, but we also do a full 2.5-hour neuropsych assessment, and then they're assessed at a consensus meeting in neurology to confirm that they are, in fact, as best as we can tell, cognitively unimpaired. Of this sample, 157 actually turns out we now have 166 fMRI data sets, and here's some data on breakdown there. I want to stress again, while Beth and I sort of co-lead SAMS, really there's many leaders in SAMS. Many folks have driven the science forward. I want to acknowledge Ali Trell, who was a postdoc for the first few years of her time here and overlapping with PHIND, and I would say that the support from PHIND is what actually helped propel Ali into an instructor position here as well in neurology. All right, so just a cartoon again, just so we know what sort of measure we're talking about. Boy, that's counting down, not up. That's not helpful. Okay, great. Here's one mechanism that we're going to focus on for the rest of the talk, this phenomenon of cortical reinstatement. During an event, again, the various features of the event are represented in cortical and subcortical structures. Their outputs from these regions ultimately propagate into the hippocampus, and the hippocampus, through its learning mechanisms, builds through a population-level code, sort of a conjunctive sort of memory representation that is sort of capturing the co-occurrence of those features, such that at some point in the future, you get a cue that's sort of one part of that past event. It propagates into the hippocampus and triggers pattern completion of that hippocampal ensemble, and then the hippocampus is thought to drive information, representations of the other features back out in the cortex such that you can consciously remember that other thing that co-occurred with the cue at that moment in the past. And so we call this sort of cortical reinstatement, we call this phenomenon cortical reinstatement. All right. So you can measure, when I was a grad student, we started doing fMRI way back when. We would collect one slice in the frontal lobe, and that would be like a 7-minute scan, and we'd move the slice, and we'd collect another slice, and that was the entirety. Here we can do whole brain imaging. We can take the data at individual trials, feed it into a machine learning classifier. We can know what kind of features are being represented in that individual's brain at that moment during this event, and then we can measure whether those features are reinstated during individual retrieval trials. So in this study, subjects are encountering individual words paired with famous faces or famous places. We take the patterns from the ventral visual cortex, regions that represent sort of visual objects and representations of scenes, and we feed in those patterns to a classifier in order to train the classifier to sort of detect patterns associated with the representations of faces or places. At retrieval, subjects encounter individual words now, and they're trying to remember, gee, did a face go with that stimulus previously? Did I see a place with it? Do I recognize the item as old, but I don't remember what co-occurred with it? Do I believe it's new? And so you can take these retrieval patterns, feed it into the encoding train classifier, and essentially quantify the extent to which the brain evidence at retrieval its similarity to the brain evidence during learning. And this is our measure of cortical reinstatement. In the SAM sample, as in the broader the broad literature, older adults performed worse on this associative memory task. Importantly, older adults also demonstrated weaker cortical reinstatement. And this is the case even when they were successful in remembering, being able to tell us what they encountered. These are all successful trials, and these are sort of means for a given individual. Even when you're remembering and you're an older adult, the strength of that evidence is weaker, okay? Importantly, controlling for age, sex, and education in the SAM sample, reinstatement strength predicts. So individual differences in the strength of cortical reinstatement predicts individual differences in memory performance. This is in the scanner, predicts performance on the tasks in which the brain data are being collected, but it also predicts performance in out of task sample. That is, the brain data in the scanner predicts neuropsych scores at the individual subject level. All right. I think I can almost do this in 5 minutes. All right. So as noted, samples of cognitively unimpaired older adults. Some of those individuals are going to be amyloid positive, others are going to be amyloid negative. That's true also in SAMs. We identified via CSF, and for a small subset where we didn't have CSF from amyloid PET, those who are amyloid positive or negative. And then from CSF, we also measured p-tau 181, and as already noted, those subjects who demonstrated elevated p-tau, which tended to be the subjects who were amyloid positive, which is consistent with the literature, they're the ones who demonstrated worse overall episodic memory. In SAMs, we see again what's out there in the literature. E4 carriers demonstrate elevated amyloid, and they also demonstrate elevated tau, in particular in individuals who are amyloid positive. All right, so here are the questions I started off with, teased you with. How does APOE genotype relate to cortical reinstatement? What about amyloid and tau? And do cortical reinstatement and these AD biomarkers explain unique memory variants? So, just a few data points here. I won't overwhelm you if I haven't already. E4 carriers demonstrated weaker cortical reinstatement in multiple brain regions. Individuals who are amyloid positive also demonstrated weaker cortical reinstatement in multiple brain regions, and ditto for individuals who were tau positive. And so each of these risk factors or measures of preclinical AD were actually associated with weaker neural mechanisms that underlie remembering past events. Strikingly, and we have a lot more to do here, but the effect or the relationship between E4 and the decline in cortical reinstatement in some regions was independent of these AD processes, independent of amyloid and tau, perhaps suggesting an AD-independent pathway that's impacting neural mechanisms of memory, whereas others were explained by or accounted for by amyloid and tau. All right. Last thing, okay, I'm actually doing okay. Last data point before we push on the PHIND front. The data that I've described in terms of amyloid and tau, they're global measures from largely CSF and plasma. But there's, of course, regional burden, regional accumulation that's playing out over the course of the disease. And here's some data from Keith Johnson and colleagues that Beth very nicely identified and highlighted the overlap with where we're seeing cortical reinstatement when people are remembering past events. And what you can see is the regions that we observe cortical reinstatement in the SAM sample, and that seem to relate to memory behavior, they're also early sites of tau accumulation in cognitively unimpaired older adults. And one sort of benefit that we have from the PHIND study is sort of encouraging our movement to add tau PET to the SAMS population. So here's a couple of participants from a paper from Beth's group, not the SAMS participants. Both of them are healthy, cognitively unimpaired, healthy older adults. And you can see that they differ in the accumulation of tau in the ventral temporal cortex. And what we observe in SAMS, it's only 22 participants. Do not put any weight in this. I believe we need to be many tens of subjects more than this, but we're collecting data actively right now, and so we expect to be able to get this number up to about 120 or so. Preliminary results suggest that regional tau burden in the ventral temporal cortex actually relates to the strength, the ability of an individual to represent event features during an experience, as well as the ability to reinstate those event features later on. And this, again, relates to associative memory performance. All right. I'm going to skip beyond this other than to make the point that with SAMS-1, we've got multiple features. Not only, age is a predictor of episodic memory, but we've got these functional measures, and this is just one of them, a cortical reinstatement that is related to memory. We have these AD biomarkers, in particular p-tau, and all of these are making independent predictors or explaining unique variants in individual differences in episodic memory performance. Okay, so that's this point, which is there appear to be multiple pathways related to aging and AD that simultaneously influence this sort of neural mechanism of memory. It's just one of multiple neural mechanisms that we're interested in. Nowhere have I said anything about prediction. Nowhere have I said anything about sort of precision health. And this is where PHIND comes in, right? So PHIND, sort of the call for proposals for dream teams. Interested in one category was biomarker research to study the molecules that are indicative of health and disease, to predict risk for disease, and to bring together interdisciplinary groups. In SAMS, we've got this remarkably and uniquely deeply phenotyped population. I know of no study, I think Beth, we know of no study that has the functional measures that SAMS has in terms of learning mechanisms and retrieval mechanisms, along with high field, high resolution structural measures combined now with these molecular measures. And so we've got a deeply phenotyped population, and thanks to PHIND, we've extended it to actually a longitudinal design that's going to allow us to develop predictive models. And it's also allowing us to collaborate with numerous folks on campus. So here is where we had this sort of support from the NIH for the baseline study, just this cross-sectional study. We got a little bit of support from, grateful to Tsai Neuroscience Institute to allow us to add some tau PET imaging for the last few tens of subjects. PHIND came along at the perfect time, sans sort of COVID. That actually allowed us to begin to extend SAMS into a longitudinal design. We began to bring folks back at 3.5 years out in what was dubbed at the time visit 2. It's now visit 1.5. We got a few tens of subjects in at this prior to COVID. Thanks to the leadership team, we hadn't initially thought to collect CSF again. We were encouraged to collect CSF. The CSF data that we've been collecting, we collected through this wave, along with other measures, have already been in numerous publications, not just from our group, but from others here on campus. We have abstracts going out to the Society for Neuroscience using some of the structural measures collected. Just this week they're going out, so we're very grateful. But of course, COVID hit. We didn't get to the sample of 60. I'm going to wrap up. One slide left, I promise. We didn't get to the full sample of 60 we were hoping with in PHIND, but we did get to tens of subjects, which was fundamental, critical data to demonstrate feasibility that we can actually bring folks back. And so now we've launched SAMS-2, which is a 7-year longitudinal follow-up that repeats everything that we did at Wave 1. We're a year and a bit into this. We've already had tens of subjects who've come back, and we've also added regional tau, PET imaging, which also then gets us resting state, fMRI, DTI, et cetera. So we're very excited about what ultimately is going to come from PHIND's support and investment. I don't think we would have gotten here without PHIND. Okay. I've already alluded to the numerous sort of ways in which the data that we've collected are being leveraged by others, so I'm just going to end with, again, noting I'm just up here. I'm a cognitive neuroscientist. I babbled on a lot about memory mechanisms because those are things I know. I'm not an expert in Alzheimer's disease, so I'll do my best in fielding questions. This study is a collection of experts, and I just want to, again, acknowledge Beth, acknowledge Ali Trell, thank the folks over here on the left who are part of the SAMS team, and thank PHIND again, and thank everybody for your attention.
Some really impressive work from everybody, so thanks so much for sharing that with us. And so I'm kind of wondering, looking through to the future, what do you think is achievable now, or could be in the near future, given what you've accomplished over the last several years? You've highlighted a lot of really interesting data, so I'm wondering how you can leverage that kind of moving forward. So I'll start with you, Dalia.
So I think there's two things that that we've learned that we want to move forward. One is that we are complex beings, and we cannot just focus on blood sugar. We cannot just focus on what food affects what blood sugar, which was our original thought. Like, no, with what are you eating it, after doing what, after sleeping how, and we probably will start to look at stress levels and all of these things. So we're complex people, and that's why we want to do this next study, where we're collecting information about the whole thing, the whole human experience. It's a little mighty goal. The other thing is that we can't just rely on going to the doctor once or every couple years and just getting that data point and seeing how things are going, because some of these things we can if we could measure them more often, we can probably catch them earlier. And so there's a lot of work in the lab on trying to do all of these more intermediate measurements and understanding, because truly each one of us is more similar to ourselves even when we're sick than to another person. So me healthy and me sick, I'm more similar to me than me healthy and you healthy, or me sick and you sick. So understanding how myself I'm changing is better when I have a little more data about myself and catching it earlier. So I think that those are where we're going with, more measurements and more densely measured.
Excellent. Ian?
I'm going to make a push for the environment and study the environment. That all of that is true, and obviously wearables in the session before, and skin sensors and sweat sensors, and all of those provide more data. I'm going to put out there that I think climate is changing. And we have now national databases on particulate matter across the entire country. And if we can tie people's health, brain function, cortisol levels to their census tract, right, address, we've already been finding in this PHIND sample that we can predict levels of depression from the timing of exposure to air pollution and water pollution. And we've seen lead and arsenic in water, even in the Bay Area, being related to levels of anxiety in our kids. And so trying to integrate, and you can care or not care about the environment, it doesn't have to be the environment, but the more domains of a person's life you can integrate to try to predict health and the onset of disease, I think the stronger position we're going to be in. And we have that capability now.
Absolutely. And Anthony?
Great. I'm going to give a two-pronged response. The first is one sort of what's been eye-opening for me. So again, as a cognitive neuroscientist who really hasn't spent much of my career up until recently focus on AD-related processes, and then having opportunities to collaborate with an amazing sort of set of folks here on campus, I've been struck by how rapidly the biofluids field is changing, and the number of markers, new markers, be it immune system response to sort of blood plasma sort of measures of amyloid and tau, et cetera. It seems like that's one area of explosion that's going to be happening. It's one area that's likely, if it's not already, impacting clinical practice in terms of measures. You can imagine that sort of coming at some point. And so that's been eye-opening for me, and really kind of rewarding to see the way in which a large project like this, the effort put into collecting the data, then through sharing and through collaboration enables discovery science that you'd never conceived of. The second then is you know, and it may well be it's because it's the closest thing to, you know, in terms of, you know, where I've spent much of my career. I'm excited about these functional measures, believe that once we've cashed out the sample at a 7-year follow-up, but that's still going to take a number of years, that there's going to be unique predictive information at time 1 that, you know, is going to be valuable in terms of understanding both disease-related, at least AD-related, as well as AD-independent sort of change in memory performance over that time in clinical status. And that's then going to interact with, so how do you make that scalable? And there is, you know, with scalp EEG, you don't have the same sensitivity to these sort of cortical signals that I've described that you have with fMRI. But we're doing some studies there now, and I'm hopeful that methods that are sort of more portable, you know, scalable, scalp EEG, or there's new sort of MEG technology that is sort of going to be much cheaper, much more likely to, you know, be positioned in numerous sort of facilities. That using these new imaging techniques, assuming that my bet is right or our hypothesis is right, that these sort of functional measures really are going to be informative, the emergence of new imaging techniques might actually make it scalable.
Excellent. So I guess with the limited amount of time, I want to make sure our audience has a chance to ask as well. So I'd like to open up for any questions anyone may have.
Jack Mormon. Thank you. Those were excellent and lots of things that I certainly wasn't aware of. Professor Wagner, so your studies in the MRI weren't blinded. The patients or participants knew memory was going to be tested, and some people use memory tricks in the first place, silly images, things like that. Did that affect the results any?
So that's a great question. So the first thing, so we haven't done a study where encoding was incidental. That'll be kind of hard to pull off with this age sample, given the number of stimuli we need to actually do the science. What I will note is it's possible at the trial level, a given individual is you know, using a strategy sometimes, but not always. But at the trial level, the same signals that I described here in terms of the strength of cortical reinstatement, in the SAM sample as well as in younger adults, those signals explain moment to moment whether or not the person is remembering, explains subjective reports of how vivid, how confident they are in their memory. It explains how quickly they can express their memory, their reaction time. Some of that variance may be related to some of these strategic processes that you're describing, attentional processes. But I suspect that that's not the whole story. While I anchored last sort of, and then so we can move on, while I've anchored on the hippocampus, the mechanisms within the hippocampus and interaction with the cortex, my lab, we spend a lot of our time thinking about how it is that our goals during an experience, during learning, as well as our goals when trying to remember, interact with these same memory mechanisms, interact with our attentional systems. And built into PHIND 2, we're getting assays of attention, we're getting assays, in some cases, of goal states. We're getting measures of these frontal parietal networks that support those kinds of mechanisms. And so that is an open question for us. We're very interested in that. I suspect there will be significant variance explained by engagement of those networks, but that's all to come. Thank you.
Maybe you can email me some of those tips.
I'll try and remember to.
Yeah, thank you.
Hi, thank you very much for all of your talks and for all the talks today. To Dr. Gotlib, the sort of two 800-pound gorillas in the room are screen time and social media. And that affects all of us, not just adolescents. You have a few thoughts on that?
Yeah, let's just call that one 1,600-pound gorilla, right? That I think absolutely social media use is critical in a lot of this. I just have to say that these trends were evident long before social media was a thing. So I think that they have exacerbated what we've seen as already increasing levels of depression, of distress. I think the really high levels now no doubt, and we've all read stories of the bullying and the suicides. That's absolutely a part of it, but I just want to be clear that this was already a process in motion.
Excellent data, Dr. Gottlieb. As you all know, the stressors do cause downstream effects that us adult doctors are concerned with, particularly cancers, cervical cancers, one in particular that's associated with early life stresses, as is cardiovascular disease. I'm just wondering, should these now adolescents be really encouraged and warned about this? Certainly they should be getting their HPV vaccination. Certainly they should be getting good clinical follow-up. And you have a great deal of data and contact with these patients to really make an impact. So thank you for your great work.
Thank you. And I'll just quickly say, yes, absolutely. That I think that our focus on depression and suicide, we could have focused on almost any adverse outcome, including physical alterations. And I talked with Anthony a lot about, are we seeing early signs of, I think older adults are too late to see early signs of Alzheimer's. I bet we're seeing it in kids, and we just, but it takes such a longitudinal study to do that, and to do what you're suggesting too.
We know, for example, C-reactive protein can
Yeah.
be a predictor. And so you're measuring stress, and that's a nice thing to know.
I completely agree with you, and I hope that I'm here in 30 years when these kids are, 50, and we can start to look at physical functioning. That's not going to happen.
Well, I guess one question I had for you, Ian had some really interesting data of like pre and I guess peri was the term, pandemic. If you think about diet, I remember seeing a lot of, articles you see, you get really buffed and then you get really out of shape and then, kind of go through these cycles. How does that factor into your precision diet work? And, do you have any interesting findings, kind of like what Ian had?
It was interesting because we had to, like everyone else, stop our study, like hard stop. And some of them were in the middle of that phenotyping phase. So they had done just one test, but not another. And so we were calling them back once we were reopening. And some of these people, it was interesting, we had to repeat everything because their bodies were different. Like, no, we are not the same person that we phenotyped before the pandemic, because some of them had gained a lot of weight, some of them had lost a lot of weight. Because, I had a lot more time to walk and a lot more time to cook, and I just decided this is my time to get fit. Like, fantastic. So, but we saw those two. Some people just became much worse in terms of their metabolic health. Some people became much better. So it was interesting to see.
I'm curious for you as well, Anthony. Because I think part of Ian's was structural and functional MRI work. So
Yeah, so as this discussion was unfolding, I've been making a note for myself to follow up on this in our sample. So through collaboration with Hamilton O and Tony Wiscori's group, they've been using sort of protein measures. They have a paper in which the SAMS data is part of the sample, focusing on organ aging. And organ aging is a function of sort of, measuring particular proteins that are associated with particular organs, as well as then the structural measures along the lines that Ian sort of described in terms of being able to calculate a brain age and whatnot. And I think in SAMS, we're positioned to have these data 7 years later. It's going to be post-COVID. There's going to be a lot of things that have changed, but to be able to identify individuals who over that 7-year period might have shown, faster organ aging rates for particular organs, perhaps including the brain, off of the protein measures as well as then the structural measures as well. And won't have an ability necessarily to tie it back to stress reaction to sort of COVID and other things along those lines. But we will have sort of that test-retest ability in terms of, Ian, brainstorm for us.
Well, I bet you have same-aged people as we did before the pandemic and
absolutely.
after the pandemic, right? And even just to compare those two cohorts,
We don't have that. So we were fortunate that wave one
Was
completed the last SAMS participant enrolled in
right.
March of.
So the youngest wave 2 is older than the oldest wave 1?
Oh, no, we'll definitely have that. But,
Well,
but wave two is the same, so we'll be able to look at the change scores.
Yeah, I'm sorry, I'm
But
not
we
thinking
will also
about
be able to look at the matched.
The
for
matched age,
Yeah,
right.
well, of course, we also have, yes, we'll have
Right.
that, yep.
Great,
Great.
let's talk.
Well, great. Well, thanks so much. I think this is really an exciting panel, and it's a great build out of Sam's vision, and now building into Joe and Gary's vision. And so, really appreciate everything you're doing, and looking forward to continue to work with you in the future as well. So, really promising, exciting, longitudinal, more data. So thanks so much for all the great work, and let's keep in touch.
Thank you.
Thank you.